Good afternoon. Oh, I'm so glad to see a full house today. I'm Barbara Snyder, president of Case Western Reserve, and it is my privilege to welcome you to the Alan and Constance Ford Distinguished Lecture Series featuring today Dr. David Van Essen. Dr. Van Essen is internationally renowned for his Human Connectome project, a Herculean effort to chart long-distance neural pathways in a large population of healthy adults. In just a few minutes, you're going to hear from him. But first, I want to tell you something about the namesakes of this lecture series, Alan Ford and his late wife, Connie. Alan Ford earned an MS in Engineering Administration from the Case Institute of Technology in 1964 and served as a university trustee for a remarkable 27 years, including 4 years as chairman, and he also served as head of the university's capital campaign, the last one, which went from 1989 to 1994. He received the University Medal, Case Western Reserve's highest honor, in 1994. And we are so proud to have this as his legacy, among many others here. Alan is now an emeritus trustee, and he rarely misses a board meeting because they are so much fun. He continually braves all the weather that Cleveland can muster, whether it's hot or cold, to attend events on our campus. He is a dear friend, a trusted advisor, and a passionate advocate for our university. And I am so happy that he could join us today. Alan, thank you. Now, sadly, Connie is not with us anymore, but I do want to tell you something about her. Connie Ford received an MA degree from Western Reserve University in 1953. You see why these two better halves came together. I love it. And her passion was art. And the Connie Ford Teacher Resource Center at the Cleveland Museum of Art is a testament to her artistic passion. We love the way that both of you have supported your alma mater. In addition to this lecture series, you should know that Alan's generosity is reflected across our campus, including in the Alan and Constance Ford Professorship in the Department of Biomedical Engineering, which is held by our department chair, Bob Kirsch. The Alan Ford Visiting Distinguished Professorship Program brings to our campus renowned researchers and business leaders who interact with faculty and students and members of the community. These scholars are then invited to present the Ford Lecture, and since 2005, we have welcomed 17 scientific pioneers and successful entrepreneurs to our campus. Previous speakers include Dean Kamen, inventor of the Segway, Richard Klausner, former leader of the National Cancer Institute, and Robert Langer, the brilliant MIT scientist at the forefront of tissue engineering, transdermal drug delivery, and materials science and medicine. Today, we are delighted to have Dr. Van Essen, and gatherings such as this allow us to do what great research universities do best. Whether discussing novel approaches or debating untested theories or sharing life-changing discoveries, we do it all here. It's great to connect, to learn, and of course to grow. And that is what I think Alan and Connie intended when they set up this lecture series. What a lasting gift they have given to all of us. Here to tell you more about our speaker today is Bob Kirsch, chair of our Department of Biomedical Engineering and holder of the Ford Chair.
Wow, it's really great that you all came today. Thank you. Before I introduce our speaker, our fine speaker today, I'd also like to echo my thanks to Alan Ford. You know, I'm really honored to be the Alan H. and Constance T. Ford Professor and Chair of Biomedical Engineering, so I'm part of this namesake as well. And very fortunate, like Barbara said, to have Alan as an advisor and a mentor. We just had a nice chat the other day. In addition to my position, Alan has contributed to our department in many ways. And one of them is this lecture today. So this allows us to bring people like David Van Essen to Case. And as Barbara said, this is what great universities do. So thanks again, Alan. I'd like to quickly thank Lauren Biddlecombe for doing all the logistics to make this work. Thank you, Lauren. Anat Matabushi and Dominique Duran were the faculty organizers for this. Anat's a relatively new member of our faculty, but him and his Center for Computational Imaging and Personalized Diagnostics is really off to a roaring start. I think you'll be hearing a lot more about Anat in the future. Dominique Duran is not new to our department. However, we're really fortunate to have such a national leader on our faculty. So let's get to the business. It's a pleasure to introduce David Van Essen from Washington University. David obtained his bachelor's degree in chemistry from Caltech, PhD from Harvard. He was a postdoctoral fellow with two guys named Hubel and Wiesel at Harvard and Nobel laureates. He was on the faculty at Caltech until 1992. He moved to Washington University then and was the head of the Department of Anatomy and Neurobiology for 21 years. Wow, I'm very impressed. So he's currently the alumni endowed professor and in the Department of Anatomy and Neurobiology at Washington University. His list of accomplishments are very long. He was editor-in-chief of the Journal of Neuroscience. He was a founding chair of the Organization for Human Brain Mapping, president of the Society for Neuroscience, on the advisory boards for NINDS, HHMI, the Allen Institute for Brain Sciences, is a fellow of a number of prestigious organizations. He's a very renowned researcher. He's known for research on how the brain organizes and processes visual information. He's demonstrated that there's many areas in the brain that contribute to this, and they're connected in many hundreds of ways. He's developed new techniques to map these visual areas in people and in animals. He's developing surface-based atlases to understand the similarities and differences across the individuals. And most recently, he's been the principal investigator of the Human Connectome project. This is a $30 million effort by NIH to map the human brain, and I think we're going to hear about that today. David's work really aligns very well with what we do here in BME and what we do with our colleagues in other departments, radiology, neurosciences, and many other departments across the university. So we look forward to hearing David's perspective today. I'll let him introduce the title, but help me welcome our speaker, David.
Thank you very much, Bob, for that excellent introduction, and I appreciate the introductory remarks of President Snyder, and I'd also like to acknowledge my appreciation to our Alan Ford, the honoree of this event. So, we've got a lot to talk about, and let's jump right in. I love numbers about the brain. Each of you has about 3 pounds worth of squishy brain tissue inside your skull. It's got about 90 billion neurons for each of you. The dominant structure is the cerebral cortex. The cerebral cortex dominates in mass, 80% or so, but it's only got a minority of the neuronal count, 16 billion or so. That's because it's dominated by synaptic connections, more than 100 trillion of them, plus a lot of mileage of white matter axons going through connecting long-distance regions. We've also got a pint-sized cerebellum. It's only 10% of your brain mass, but it contains the great majority of all the neurons. And then the third major compartment is loosely called the rest of the brain, the various subcortical structures, less than 10% of what's in your brain and less than 1% of your total neuronal count, yet incredibly important for keeping you alive and well and letting you know if you're hungry or thirsty and so on. So the fundamental challenge is how do we untangle this mess, this elegant wiring of the brain? And I'd also like to put it into perspective, especially for an audience like this. There's an oft-stated comparison between brains and or versus computers. The Deep Blue became chess champion 15 years ago, and they're getting even better. We still like to think that we're a little bit smarter than many computers in some ways at least. We do our computations very differently. Computer chips now can do gigaflops of operations on tighter and tighter packing of the electrical and computational elements. We're stuck with these billions of little tiny neurons, each one of which has about 10,000 synaptic inputs and outputs. outputs onto dendrites coming from axons. And we convey information at a pace, a more leisurely pace of about 1 to 100 spikes or nerve impulses per second. So understanding computers can be done at many levels, as everyone knows. We can try to understand the architecture of the hardware. We can get into the operating system level, and most often we want to understand what applications can they do and how do they do it, how do they need to be programmed. For understanding the brain, we have fundamental challenges to decipher the wiring, the connections, as well as the signals that carry and process information. And then ultimately, we want to explain behavior. And I can assure you that just knowing what the wiring is and the signals is not on itself going to be sufficient to explain behavior. So that's the big picture. Let's narrow down our focus for today. It's about connectomes, but we have to choose amongst several options. A connectome is, in quotes, a comprehensive map of neuronal connections. But we have to put it in quotes because we don't have access to all spatial scales at one time. So I like to talk about the macroconnectome, which is the effort to understand long-distance connections from one patch of gray matter to another through the underlying white matter. And that can be done, as you'll see, down to a resolution of the imaging elements, the voxels that are, depending on the modality, 1 to 2 millimeters on a side. Over on the right, we see a schematization of a microconnectome concept, which is the effort to reconstruct every single neuron, glial cell, axon, dendrite, and synapse in their exquisite entirety, but only up to the level of a fraction of a millimeter cubed per date. Then there's one intermediate level that's roughly called the macroconnectome concept, which uses anatomical tracers and animal models to study connectivity locally and at long distances, but requires studies of many brains to build up a big picture. So those are the options, and we're going to focus, because of our interest in the human brain, on this macroconnectome domain. But before I get to the human connectome and the project that has driven much of the recent progress, I want to give you a fairly extended introduction about mammalian cerebral cortex, cartography, connectivity, and individual variability of human cortex, because it really informs and puts into perspective some of the challenges we face, as well as interpreting the exciting advances we're starting to see. So here's a few brains. Here's our brain, the big one in the room. At the smaller scale, we have a mouse brain that weighs in at less than a gram and has about 1,000-fold smaller size as well as neuronal count. I'll talk a fair amount about the macaque monkey brain, which is about 1/10 our size in, again, size and neuronal count. I'll say a little bit about our closest living relative, the chimpanzee. It's 1/3 the size, but it being so close to us in evolutionary relationships, the differences between us and a chimpanzee tells us about what makes us uniquely human. As you can see at a glance, all but the mouse in this quick survey are convoluted cortices, but the pattern of convolutions varies enormously. And that brings us to one of my favorite realms, that of cortical cartography. So I am a card-carrying cortical cartographer for many decades. I started out literally in the mid-70s using pencil and tracing paper to make flat maps of pieces of convoluted cortex to get the concept that we need to go beyond just taking a few slices here and there. A decade or so later, Dan Fellman and I made a flat map of the entire macaque cerebral cortex, and then we took information, evidence from the published literature to identify several dozen visual areas in color and several dozen other areas based on information about their structure or architecture, their connections, their maps of the external world, visual and somatosensory maps, as well as differences in function. And sort of this fourfold fact-finding quest led to this kind of portrayal, one version of a parcellation, but if we look closely at it, there were a couple of areas like the primary visual area V1 and this little red area MT that really passed muster cleanly by all 4 of these approaches. Most of these other areas on that version of the cortical map had subtle boundaries, and there was noise and bias in the data and discrepancies in the literature that persist to this day. So cortical parcellation, making better maps of the cerebral cortex, is way behind Earth cartography. It's a really hard problem. One of the issues that we need to cope with, especially for the highly convoluted human cerebral cortex, is to capture the shape of the cortical convolutions more accurately than pencil and tracing paper can do. And so here we're taking advantage of the free surfer segmentation algorithm to capture the shape of the outer or peel and inner or white matter surface. And then we can reconstruct that into a geometric surface model. And we can take a spin around this and see the lateral and now the medial view of the cortical folds. We can see the deep crevasses as well as the shallow wrinkles. We can take this geometry and inflate it gently and yet preserve a marker of cortical shape. The darker it is on this inflated map, the deeper it was in the original convoluted surface. And this has been done now for more than a decade, but for the Human Connectome project data, we're capitalizing on notably high spatial resolution, 0.7 millimeter voxels, and an improvement to the free surfer segmentation algorithm that more accurately captures these cortical folds. And that's really important, as we'll come to. But first, let me stick with our multi-species survey. We can take high resolution MRI, structural MRI of mice, monkeys, and human individuals and group averages, and then we can do this surface reconstruction game and use this to represent the 3D shape and the inflated shapes of cerebral cortex, and in unique reconstructions, the cerebellar surface of these species as well. And onto these maps we can portray whatever we want, and the thing I want to focus on next is to come back to cortical parcellations. The only mammalian species for which we actually have an accurate consensus map of the entire cerebral cortex is the mouse, and this is thanks to the work from Andreas Burkhalter's group in my department. And this is actually not a computerized map, this is a histological section of a physically flattened map. And you can see, for instance, the so-called whisker or barrel fields of the primary somatosensory cortex defining that area. And you can see a grand total evidence for about 40 different cortical areas. And that's how many the mouse has, to a pretty good approximation. For the macaque monkey, we've made progress since the map I showed from 1991. But the 2 decades later, we have an estimate there are about 130 cortical areas, but only a few dozen of them are consensus ones. The rest, there are contentious and appropriately unresolved debates. We just don't have enough information yet. So it's not really ground truth as it is for the mouse. And then for human cortex, we have this portrayal I show you on the right, which is the set of 52 accurately surface mapped areas, the primary and early visual areas and somatosensory and orbital frontal areas. The rest of the cortex is not terra incognita. We know a lot about it, and there have been published parcellation schemes, but they're not really satisfactory representations. So we need to fill in the blanks, and we'll come back to that. But the next thing I want to touch upon is a more closer comparison between macaque monkeys and humans to see not just that they're different, but in what regions and in what ways are they different. So on the left here, you see a morphing from a monkey to a human cerebral cortex using the subset of areas, early visual and somatosensory areas and a few others, that we are confident are evolutionary, have a common ancestor. And so they're homologed. We use them to constrain the registration from monkey to human. And then we can, on the right here, show you maps of areal expansion. The red regions are hot spots where the cortex is about 30-fold more expanded in humans than in monkeys, and other regions like the early sensory areas are only a factor of a few 2- or 3-fold expanded. So evolution has concentrated on a subset of regions that turn out to be regions, lo and behold, are heavily involved in our what we call higher cognitive functions, and we'll come back to that. There's another modality that's informative that comes from a clever idea that my graduate student Matt Glasser not only conceived of but exploited in the past several years. That is to use the structural images like the ones I showed you before of T1 weighted, the conventional, and another complementary T2 weighted image, take the ratio of them, do some fancy footwork, and basically colorize them and show that in both monkey and in human, the red regions here represent heavily myelinated patches or strips of cerebral cortex meshing with what was known from the classical literature, but now giving us an excellent whole brain mapping. And you can see that the blue regions are lightly myelinated, and those, lo and behold, correspond to these regions of cortical expansion. So the cognitive regions are also associated with a generically different architecture that is interesting to speculate what that means. I don't have time to show you, but I'll just mention that these same regions of evolutionary expansion are also regions that during postnatal development of healthy children, those are the regions that physically expand and get bigger neurons and more dendrites and synapses. So this is interesting turf. I think it becomes even more interesting when you compare the chimpanzee thrown in the middle. Matt Glasser again has helped acquire and analyze data from the chimpanzee, and we have these myelin maps for the chimpanzee, and lo and behold, the chimp is in between in terms of these lightly myelinated cognitive associated regions are larger than in the macaque, but far smaller than in the human. So the speculation that we're going to analyze more quantitatively as time allows is that it's these cognitive regions that have really super expanded in the human lineage. And so that's food for thought. And we're going to be aided in this comparison by the fact that the myelin maps give us actual candidate signatures for areas that we suspect are homologous across all species, such as the frontal eye fields and this neat little area, the MT complex. So let's move on to the next subject. I told you it would be a long introduction, but I hope it gives you a flavor of important relevant material. Connectivity. So we focus here on the macaque monkey, where that's really the primate species where we have the closest to ground truth. Again, a quick historical context. Back in the 70s, I thought the macaque wiring diagram was kind of complicated. It wasn't just a serial V1 to V2 to V3, but little did I appreciate how complex it would turn out to be. One major milestone was John Mansell's, as a graduate student in my lab, realized that we could use not just the existence of connections, but the layering of origins of cell bodies and terminations of axon synapses to distinguish forward versus feedback pathways, and let that arrange the cortical connectivity pattern known at the time into a hierarchy that then expanded into this subway chart-like pattern that's on my tie, just for future reference, and relate that to the map of cortical areas that you saw before. So while that was progress at the time, it was frustrating because when we made a connectivity matrix, so taking the same information you see in the colorful chart and making it into a 32 by 32 matrix of whether projections or connections are present between one visual area and another, all we could enter at the time was it's there or it's not. There was precious little information about the quantitative strength. Even though we knew that there were quantitative differences, we didn't have a handle on it. A decade later, my graduate student, Jim Lewis, was able to get quantitative connectivity maps for a handful of injected areas, but it wasn't large enough to build up a complete and satisfying big picture. So there's been a big step forward in the last several years, thanks to work from Henry Kennedy's lab. Markoff et al. Have published several studies in which they've taken the same concept, inject retrograde tracers into identified visual and other areas, and then painstakingly count the total number of labeled neurons and express that as a fraction of the grand total. And here, for example, you can see an area-to-area connectivity map for an injection in area V2 and see who's talking to it and how strongly by a logarithmic plot. But what was amazing from this was really two things. One is the strength of connections, when properly quantified, ranges over literally 5 orders of magnitude, from the strongest to the many medium strength to weakest. And that was true for every area, even this area up in the frontal cortex, which has even more. It has the great majority of cortical areas are sending at least some information up in that direction, but it follows the same distribution, which is a log normal characteristic that is, since it occurs in all areas, we think is of great general significance, we hope. We can take all of this information from 29 injected areas and 91 target areas and note that at this stage there's evidence now for well over 1,600 pathways just within the third of the macaque cerebral cortex that's been carefully studied. Quickly, I'll just note that the same concept applies in the mouse. We can take the mouse cortex, put tracers into there, and Andreas Burkhalter has shown that the same kind of connectivity matrix shows a smaller number of pathways overall, but this same kind of log normal distribution, again suggesting important generality. Let's now move up to human cortex and set the stage for the Human Connectome project, but first by commenting on this issue of individual variability, which I touched upon before, but now let's take a closer look. So if you look at 4 individual humans, the right hemisphere lateral views, you can see they're all highly convoluted. You can see some consistencies, but there's a lot of variability that's present between every pair of people in this room. Now let me add a twist to this. There are 2 pairs of identical twins here, and you might think, since they're genetically identical, surely their cortical folding patterns would follow the same instruction set and be nearly identical. Well, if we inflate these surfaces and look at these maps of what we call sulcal depth, I'll just cut to the chase and tell you the answer. This is a pair of twins, and yet they have very different folding patterns in some regions. Ditto for this pair of twins. And so basically, the folding patterns turn out to be modestly heritable. There is more of a correlation amongst identical twins, but it's not as much as you might have guessed. And that raises an important question, both from a methodological standpoint, but just out of sheer outright curiosity. Why is the cortex so folded? Well, it gets a lot of surface area inside your skull, but what makes it fold? And what makes it fold so differently for a dolphin or a sea lion or a camel, and why are some species not hardly folded at all? Well, a light bulb went off for me 15 years ago, and here's the storyline. So one important fact is that cortical folding in humans occurs in late gestation, the 3rd trimester, and this is just around the time when the long distance connections between different regions are forming in the white matter. One night, literally sitting on my living room couch, I was musing about this, and thinking about the part of the brain I knew the best. In the macaque, area V1 has an orderly map of the visual field, and it has powerful connections with the neighboring area V2 that preserve this topographic mapping. So the thought and the observation is there's a gyrus that folds to form these 2 regions in the adult. And the light bulb notion is that if there are such massive connections from one area to another, suppose there's a little bit of mechanical tension. Suppose each neuron is treating its axon like a fishing line, trying to reel it in after it's snagged a synaptic target here. And if everybody pulls together, they would form a gyrus in between, and a sulcus would be a loser in that kind of competition. Well, this would explain many things, including the notion that cortical folding is consistent in regions that are dominated by major pathways like V1 to V2, and variable folding could occur in these temporal and parietal and frontal regions that are this mosaic of many little balkanized areas that may be variable in their size and connectivity. So I still like this idea, but I have to be candid in admitting it's, at this point, nothing more than an attractive hypothesis. There's evidence for, evidence against it, but no compelling answer. So we have to be patient, but I'll pay $1,000 to anybody who can give me a sealed envelope with the answer. Given that variability and whatever the mechanism, the reality, especially for the Human Connectome Project, is if we want to compare one human brain with another, we have to cope with this variability. The traditional way of doing this has been to take volume images of the type you've already seen and register them to one another to form a group average and to use that as the sole basis of averaging. And that's still a good thing to do, especially for the subcortical structures which do align well. But for the cerebral cortex, because of the immense variability of these convolutions, we need really to use a surface-based approach. And one way my lab developed some years ago was to use consistently identified landmarks to register individuals to an atlas and to form a group atlas like this. That was good, but it wasn't good enough. And so we have adopted the free surfer strategy, similar in concept, but using a better registration algorithm. And it gives a cleaner group average that looks more like an individual subject. But we've made improvements on the free surfer maps by essentially aligning the left and right hemispheres to be in sharp correspondence. Again, this is good, it's progress, but it's not good enough. And the reason that I'll come back to later, but I'll just set the seed for it, is this kind of folding-based registration that uses these highly variable folds, it actually does a great job of aligning areas like V1 because they are consistently related to the underlying sulci, like the calcarine sulcus. This little patch over here is a group average, a population average probabilistic map of the area MT complex, and it's not well aligned because the areas themselves have a variable relationship to these arcane folds. So we'll come back to that, but I'll just stipulate as a general principle, folding-based registration can't possibly do a perfect or a really great job in regions that are highly variable in folding and its relationship to cortical areas. Put that on hold, and let's finally jump into the Human Connectome Project proper. I'll give you a quick overview, then a tour through the major modalities, how we get the data, and more importantly, what we're starting to learn from that. I really need to emphasize in strong terms what a hugely rewarding experience it has been to work with now a little more than 100 individuals, a really intensely dedicated, hardworking group, including my co-PI, Camille Ugerbill, and many others who've really done yeoman's work to take us where you'll soon see we've gotten. So our objectives are to study 1,200 healthy adults drawn from a population of twin pairs and their non-twin siblings to give us greatest access to studying heritability issues that I touched upon with the folding patterns, and also for the genetic analyses we'll be able to do once we do genotyping on the blood samples that are coming in. We're doing cutting-edge neuroimaging using a Skyra 3 Tesla that has a souped-up gradient insert that gives us stronger gradients for diffusion imaging. And just this month, we're going to be starting the customized studies on the 7 Tesla scanner up at Minnesota for 200 of these 1,200 subjects. I'll give you a teaser or an introduction to how we'll be able to get better data than has been conventional using advanced, what we call multiband pulse sequences, and illustrate how that helps us for diffusion imaging, resting state fMRI, as well as task fMRI. We've been acquiring and just released data from the starter kit, 10 of the first 100 subjects to be studied with MEG. We have several hours worth of behavioral data, so we have phenotyping of various personality and performance attributes that can be compared with connectivity. We're going to be doing genotyping next year. The data is already pouring out. Much of it is already available, and not just in a core dump warehouse, but in a user-friendly repository. We want this to be a resource for discovery science by the community as a whole, and a baseline for future studies of brain diseases and disorders. Where are we? We're now in the 4th year. We've got a year and a half left. We spent the 2 years, I think, honestly, quite wisely, really trying to optimize and improve every aspect of how we acquire and analyze data. So I'll touch upon the pulse sequences that we use on the improved hardware. You'll see some of the improved analysis methods and the visualization platform we have. If you're interested in gory technical details, there's 8 papers from our consortium that came out in NeuroImage last year, and you can read to your heart's content. We've now acquired data from more than 550 subjects. We're basically on a timeline that will let us get to 1,200 by the end of the project next summer. 216 subjects worth of data was released in October. 500 subjects will come later this spring. The MEG data was just released on Tuesday. And I'll say just a couple of words about MEG because we haven't done much of the analysis on that yet. But it's using a scanner at St. Louis University, and we've got its big advantage is that it acquires very high temporal resolution, millisecond scale, at the price of coarser spatial resolution. But it's already been shown from studies by our colleagues in Chieti that you can take MEG data in the resting state and identify resting state networks driven by the MEG data that correlate with resting state fMRI of the type that I'll be telling you more about shortly. And for the task MEG data, we have 3 of the 7 tasks that are carried out for fMRI. We have time in the schedule to get that from the MEG, and you can go to our website and start downloading those data this afternoon if you want. Back to the MRI, this multiband sequence acquisition that I alluded to, it's conceptually actually clear and elegant. Rather than acquiring one slice at a time, one after another to cover the whole brain, it's possible to excite the brain with a pulse sequence excitation pulse that hits up to 8 or even more slices at one moment and then interleave all the remaining slices. And to reconstruct the scene of the crime, I mean the slices of the brain, by means of taking the 32 channel receiving array, the head coil, and unalias these images and get accurate reconstructions of each slice. And now more than 100 sites around the world are using this kind of technology to improve structural and functional MRI acquisition, diffusion imaging and functional MRI. So speaking of diffusion imaging, let's jump right into that modality. Here's an image of an MRI slice of the white matter in a T1 weighted image. Here's a classical bulk dissection to show that it's been known for really, I think, a century or so, that the white matter indeed has this spaghetti-like bundling into coherent fiber bundles. That's the good news. The challenge is that each of these fiber bundles has about several 100,000 axons running within it, and they are not all running in parallel. There are countless crossing fibers already hinted at here, but I remind you of the thousands of pathways that the monkey's brain has, and surely we have even more, so we've got to expect a lot of complexity in this underlying white matter. How do we tease it apart? Diffusion imaging fundamentally relies on the fact that water diffuses more rapidly along the length of axons, whatever their diameter, and one can use the strategies of diffusion imaging to estimate not only the dominant orientation of the fiber bundles based on preferred diffusion, but if you excite in separate acquisitions many different angles, one can get high angular resolution diffusion imaging and reconstruct in ways that estimate the 2 or 3 most dominant fiber bundle orientations, whether they're intersecting at right angles or obliquely. That's been around for more than a decade now. What the Human Connectome project is adding is a stronger gradient strength, as I mentioned. This multiband factor, we can get a factor of 3. Those two together let us get 1.25 millimeters, which is about as good as can be done these days with diffusion imaging for in vivo humans. And we have better methods of estimating the fiber bundles and in each voxel and computing the overall trajectories using an approach called probabilistic tractography. When that is applied to subcortical projections associated with a seed up in the motor cortex, this is an illustration of the improvements that are made. So on the left is conventional scanning at 2 millimeter voxel resolution, and you can see evidence for subcortical projections, but it's only on the Connectome project high resolution scans that you can see 4 discrete fiber bundles representing known pathways, or at least known in the monkey and expected to be in these regions in humans. So that's evidence that this is really benefiting our ability to look at long distance pathways. We can also use this approach to connect the dots from one piece of gray matter to another. So if we use a seed region in the inferior frontal gyrus up in the frontal lobe and take a coronal slice through that, we can see the MRI structural volume and the white matter orientation. We can zoom that up. And for every one of these white matter voxels, we can estimate the diffusion imaging-based fiber orientation, and then we can apply tractography to this, the probabilistic approach, and take a seed region here and say if we do this probabilistically thousands of times over, we estimate an overall fiber trajectory within this slice. But of course these trajectories are going in 3 dimensions, so with our Connectome workbench visualization platform we can reconstruct the 3-dimensional trajectory associated with this one seed region that we've chosen. That's hard to see in 2 dimensions, so we can rock the boat here and give you a better impression of the 3-dimensionality of this trajectory going through the white matter and eventually terminating some of these in other gray matter regions. So that's illustrated here. We take this is a slightly different seed location, and we can trace the connections through these streamlines up to their terminations in other distant regions. And then we can estimate the overall density of connectivity associated with this particular seed region, and you can see there's evidence for local and long distance connections in an individual and in group averages. That's the good news. It's encouraging, but there are also warning signs that I want to spend a moment elaborating on. So you can see these green patterns tend to occur on gyral crowns, and the deeper sulci tend to be darker, weaker. And that points to a really significant but underappreciated methodological limitation to current tractography and diffusion imaging. So if we take a gyral slice, we zoom up on this gyral slice and we see a white matter blade, and what we would like to think is that the fibers are streaming and pulling off and feeding connections all along here. But diffusion imaging, because the voxels are relatively coarse, is going to mainly capture the streamlines that terminate up on gyral crowns. That's what one might predict, and that's what in fact we see in both human cortex and in monkey cortex. I regret to report there's a paper in the literature that states this is a fact of anatomical connectivity in the macaque, but that doesn't mesh with what I've already shown you about actual ground truth connectivity in the macaque. And so we think this is an unfortunate, but deal with it. It's a real characteristic of the tractography method. It may be possible to get even better data, though the Connectome project data is pretty good. It may be possible to cope with this by better modeling, but in the meantime, anyone who does diffusion imaging, I would caution to understand these limitations and don't jump to over-interpretations without careful attention to the realities neuroanatomically and methodologically. Let me jump over to the next modality, functional MRI, and I'll start with the one that people are most familiar with, task-evoked fMRI. So in our Connectome project, we have 7 different tasks chosen to give us broad coverage of different functional systems and as much of the brain as we could get. I'll show you quickly results from 3 of them. So one of them, the subjects are sitting in the scanner, and for a while they listen to short stories, and for a while they do math math problems and tell us the answer. Another one that's cute is they do a social cognition task where they look at these images and tell us, are these interacting socially or just moving randomly when that should be in motion. And the third task I'll quickly illustrate is a working memory task. Look at a bunch of images and remember which ones you've already seen. We visualize this using an improved way of getting the big picture with this anatomical substrate. So here on the left 4 panels you can see the left and right cerebral hemispheres inflated so you can see into the folds and looking at lateral and medial views. Then we can show you the subcortical volume slices and also a cerebellar surface flat map. Now we can paint onto that the first of those tasks. So if the participants are listening to stories, it activates regions that are associated with language systems, but a very complex pattern. And in the next task, that's the social cognition one, you see another complex pattern that's not completely independent. I'll go back to the hearing stories, social cognition. There's some overlap and a lot of distinct activations in the cerebral cortex and in cerebellar cortex. Similarly, for working memory, we activate a different but somewhat overlapping regions of frontal and parietal cortex especially, lots of cerebellum. So we don't have time to dive down into the details. The general and important picture is that different tasks of the type that we've chosen engage very complex and partially overlapping networks whose organization we aim to decipher. And we're going to learn more about that by looking at resting state functional MRI. So this is 40 seconds in the life of one subject in the scanner. The bold signal fluctuates in time, and it fluctuates in a spatially very interesting way. There are coherences and correlations and anti-correlations. And if we take that, we can analyze it systematically, as shown here, by taking at each pair of locations, look at the time course and ask, are they correlated, anti-correlated, or uncorrelated? When we do that for all 90,000 by 90,000 surface vertices and gray matter voxels, we get what we call a dense functional connectome that is based on these correlations, their magnitude. We can take one row at a time of this matrix and portray for a given seed location where is the functional connectivity locally and at long distances. That spatial pattern is a map of functional connectivity for one location or another, and we can do that to our heart's content to explore these brain circuits. One of the issues that comes up, though, is if we want to do this in a population, we need to get the surfaces as aligned as accurately as possible. And I already told you that's a challenge. This is a blow-up of what I showed you before. Conventional folding-based registration does fine for calcarine cortex in V1, does poorly over here. Can we do better? The answer, the good news is, there's a new algorithm, multimodal surface matching, generated by Evan Robinson and Mark Jenkinson at Oxford and powered through and applied to the Connectome project data by my student Matt Glasser. And I'm going to show you a complicated but I think rather cool set of images that drive this point home. So here is the Connectome project data from one subject. This is the map of the folding pattern for that subject. This is the myelin map for this subject, a little bit noisier, but you can see interesting hot spots. And you can see that when we look at the functional connectivity map, seeding one of these hotspots shows hotspots that are lined up with these hotspots of myelin. And when we do the task math versus story contrast in this subject, we see an interesting pattern. Now we do this in another subject, and we see similarities, hotspots of myelin, hotspots of functional connectivity, complex functional activation, but they don't look quite well aligned. And indeed, when we align them just based on the folding pattern alone, we get a group average myelin map that's a bit fuzzy, and a group average functional and connectivity and task activation that are fuzzier than we would like. But when we add in the multimodal surface matching that's driven by the myelin maps and the functional connectivity, we get a better outcome. And that's shown down in this bottom slide. The myelin maps are better aligned. The functional connectivity and task activations are crisper. And I'll go back, fuzzy, forward, crisp, and that's because we've pushed these regions around to be in better alignment. So that's proof of concept that we're excited by. Now, how are we proceeding to make better use of these data? We want to explore further what we're learning and make these data freely available. So here you can see an exemplar of what we call resting state network nodes that are patches of cortex that have a common functional signature. They are strongly correlated in the resting state fMRI across all these locations, across a population of individuals. And you can see they're related to anatomical subdivisions like this early visual pattern here, but they don't cover an entire visual area, just a portion of it. We can use this information to make a comprehensive map of functional connectivity for 98 by 98 nodes or independent components. And this pretty picture shows in the upper left the strength of the correlations when we do what's called the full correlation map. They're strongly correlated for some, anti-correlated for others, and they can be grouped into functional and geographic subdivisions. We can also take this and regress out the common components and analyze what Steve Smith likes to call the partial correlation matrix, that is more what's distinctive to each, after regression, one component versus another, what is their residual interaction. And we can do that not only for the group average, which is what I've shown you before. Now we've inverted this functional connectivity matrix. We focus on the partial correlation, and now we can apply what's known as dual regression to get a partial correlation matrix for each individual subject. It's noisier, but it's informative about the pattern of functional connectivity for that individual. We can take all of those pairwise edges, stretch them out 4,700 long, and stack them up for each individual. The fact that functional connectivity is similar across the individuals is indicated by these vertical stripes here. The fact that patterns are not identical is evidence for individual variability. And then we can take this very informative matrix of edges by individuals and start correlating it with other behavioral measures. And I'll show you one example. If we use the functional connectivity for each individual to try to estimate the fluid intelligence that we've independently measured in each individual, we find there is indeed a significant correlation. It's not so perfect that you can look at the functional connectivity and state the exact intelligence of each individual, but it's going in the right direction, and it allows us not just to predict global intelligence, but really to start exploring what circuits, what components between a frontal lobe node and a hippocampal node. I can tell you that that is the strongest component, the strongest edge predicting the functional connectivity and the intelligence, and we can do that for the second strongest and see another spatial pattern. This is just a teaser for the kinds of relationships between brain circuits and behavior that is now open for business for the entire community to explore in healthy adults for the Connectome project data. And we hope, of course, this has relevance for the clinical realm, but that's to be determined. The last example in this realm that I'll quickly mention is the heritability. We can take the functional connectivity matrix for each individual and correlate all possible pairs, but then group them into the functional connectivity for the monozygotic twins and show that they are more strongly correlated with each twin pair than the dizygotic twins, and much more correlated than unrelated individuals. So that's telling us basically that there is indeed heritability to functional connectivity, and we can again just not express the total correlation, but start exploring what components, such as this relationship between the default mode network and a component of the language network, those are the most heritable connections or edges based on this analysis. Very early going, but as I say, we're having fun doing it, and you guys can, the community can join in this because we're sharing the data already. The Human Genome Project drove the point home that the field will advance, progress will be greatly accelerated by data sharing, and that's our vision and motif too. You can go to a Connectome DB, and after you sign the data use terms, it's not complicated. You can start downloading the data in batches for as many subjects as you're interested in, or you can get Connectome in a box and get the entire package delivered to your institution. So we've already released the imaging data, provided multiple levels of access. Importantly, we're not just saying, here's the raw data, you go through the same hard work we did. We encourage you to extract and make use of the extensively processed data sets, either in the minimal preprocessing or the denoise fMRI and the task fMRI data. Some of the group average data is already available, and there are data use terms appropriate for getting what kinds of data you want. So what has this gotten us to? We're in still the early stages of a tremendous challenge for the rest of the century, if not the millennium. The Human Connectome Project is already starting to elucidate brain connectivity and how it varies in healthy adults. There will be ongoing discoveries and insights, but we're not going to get a complete connectome, no matter what the blogosphere tells us. There are some things that really are outside our grasp, so we need to manage our expectation. We need to be optimistic, but maintain a critical eye on all aspects of the data. And we need to be optimistic, or I certainly am optimistic, that this kind of approach extended to future studies of diseases and development will aid in better diagnosis and treatment. Again, I thank a fabulous group of investigators, particularly Camille Ugerbil and my talented graduate student Matt Glasser, and of course the NIH Blueprint for funding this major effort. And I'll close with an expansion on an analogy I already made. There have been revolutions in cartography for both the Earth's surface and the brain. It started with classical maps, quaint, but we knew they were inaccurate. Book atlases made a big improvement for both the brain and the Earth's surface. Satellite imaging and neuroimaging expanded the horizons immensely for the brain and the Earth. And now the new technology of Google Earth, of course, has transformed our daily lives. And we hope that the burgeoning realm of connectomics will transform our understanding of the brain. So thank you for your attention. Any questions? Yes?
Very exciting. If I understand it well, you're establishing maps for conscious processes. Do you also, what can you conclude on subconscious processes?
Our maps are in some ways not specific as to whether the processing is conscious. Well, the subjects are conscious in the scanner even while they're in the so-called resting state. Occasionally they do fall asleep, but we try to discourage that. So in that sense, the marker we have from the functional MRI relies on signals in the conscious state. It's been shown that functional connectivity persists and is qualitatively similar but quantitatively weaker in the anesthetized state or the sleeping state. So we don't think we do think that these maps are relevant for understanding brain function at all levels, but we don't have direct access to that in the data from the Human Connectome Project. Other studies have been exploring that important issue. I'm not sure that fully answered it, but maybe you can ask me afterwards.
Well, I imagine the subject, when it's in the MRI, may suddenly get other impulses. For example, an erotic impulse or anything like that. Can you pick that up?
We do not have control. When we tell them to be in the resting state, they could be doing anything, thinking anything. All we do is ask them not to move. And so it is a limitation of resting state fMRI that you do not have independent control. And it has been criticized for being limited in that respect. What I would argue is we need all of these modalities: functional MRI, resting state, task evoked, structural MRI, MEG. You know, none of them is going to give us the full answer. Even the combination is not going to give us as many answers as we would like. But if you say, where are we now compared to 5 or 10 years ago, it is impressive and exciting. That would be the message.
With respect particularly to the functional, the resting state functional MRI, David Z and his colleagues at Hopkins have shown that particularly in the 7T magnet, but even in the 3T magnet, because there is ions in the endolymph, Lorentz forces are applied, and so it's not truly a resting state, it's a sustained vestibular stimulus. Patients often complain they're dizzy when they go on the scanner. Have you taken account of that?
We are aware of it. I've seen that. I'm fascinated by it. And I would agree that resting state functional connectivity is a misnomer at many levels. It's not truly a resting state. They're being stimulated by the vestibular system. They're being stimulated by the fact that we have a fixation spot and we ask them to fixate so that, because if we ask them to close their eyes, many will go to sleep. And it's not functional connectivity in the way that it's true anatomical connectivity. It should be functional correlation, but the field had kind of consolidated on that term before our project came into play. So this is part of what I would strongly urge everyone to understand: what are the limitations in the real data and in the interpretation. So I don't disagree with your statement. It's just the realities of understanding the full context of these data sets and how they should be carefully interpreted and avoid misinterpreting. If you see something interesting about the vestibular system in the resting state functional connectivity, you should be mindful that it's being stimulated. I agree.
I was thinking about the smaller scale connectivity in contrast with the interregional connectivity that you've been talking about, and thinking about Doris Tsau's wonderful talk at SFN. And she showed how one area, like a face recognition area, is really a constellation of 5 or 6 areas that do very different things that together make face recognition possible. Do you think that's going to be replicated throughout the brain, or is that just because that area sits at the top of the hierarchy?
I'll pay another $1,000 if someone, or $10,000 if someone will deliver the answer. That's a really important but hard problem or set of problems. So we have, as part of our task activation, part of the working memory task has faces, and we can actually see face activation patches in our data set. They don't actually show as crisp a patchiness as what Doris and others have shown, and that may be a limitation in the sensitivity of our paradigm, but we do have access to exploring the functional connectivity of those regions compared to others. My general feeling, and my student Matt Glasser, his thesis is on parcellating the human cortex. I think it's very clear that some regions, like the face patches and others, are very crisp and very tight and patchy, and we need the best resolution we can. Some of these other regions that are involved in cognition, they look to be fuzzier, but is that because we just don't have the right tools and we still haven't aligned them well enough? Or is it because they evolved very recently and their circuitry is interestingly different in ways we are going to struggle to decipher? And it won't surprise me if it's the latter, but there's still a whole lot more we can learn that will sharpen our understanding, give us better maps. And I use the analogy that our current maps are more like, at best, 18th century Earth maps. If we can get up to 19th century, that will still be a lot of progress. We're not going to get up to Google map type understanding of the human brain with the methods currently at hand, and we have to acknowledge that.
I know you spent a lot of time in your lab talking about or investigating neural circuitry at the physiological level, and I wonder if you could comment on sort of limitations or insights you could obtain from the connectome at the circuit level, in terms of how the brain works, as opposed to how it's connected.
I can comment on it. I cannot give answers to it. It's another of the really hard problems. And fundamentally my answer is to step back and just remind everybody that the good news for neuroscience is there's a lot of talented investigators working in many different realms, chipping away and making progress on many fronts. And some of these issues, we will not answer questions of how the brain computes from just what the Human Connectome Project offers. We need the animal models, the monkey and mouse models, to say what are those cortical circuits actually doing. And then to synthesize and pull it together, we need a computational strategy for integrating the information processing that we can decipher in the animal models and put them into the context of how could that be simulated in a meaningful way for the human brain. I personally am very excited about that opportunity for computational neuroscience, but I also think it will take a different strategy than anyone is currently crisply articulating. I think there are seeds of more powerful strategies, and I'm not going to dwell on it. I just think it's an exciting problem, and especially for a biomedical engineering audience, this is another great frontier, but it's going to take breaking the bonds of our current too narrow mindset for how to model and interpret brain circuits and brain function.
From an anatomical standpoint, it's pretty clear that if you were to find something that was aberrant in terms of connectivity, you could surgically go in and disconnect something. But from a functional connectivity standpoint, or anatomically to connect instead of disconnect, how do you take the next step to take this information and clinically act on it when you find out that you have a connection you don't want, or you do have a connection?
Another great but deeper question than I can properly answer, but I can comment on it. So, I touched upon it that we want this kind of approach to inform us about brain diseases and disorders. There's already a lot of evidence published out there that brain circuits are abnormal in autism and schizophrenia and so on. So we're not the first to bring that into play. But I think there are limitations to the current methodology, and they're not going to be solved, but they're going to be improved by using the kinds of analysis and acquisition and analysis strategies the Connectome project has brought to the table. What will we learn? We will learn, evidence for, some of it will be kind of global circuit non-optimal functions. Some of it may be regional. Are we going to learn how to, perturb brain circuits in ways to address the clinical circuit disorders? That's in the way the hope, and one of the hopes is that neuroimaging may provide us a metric for evaluating. If we could identify not just the autistic brain abnormality, but the spectrum of circuit abnormalities that are relevant to individual subjects, and then learn how treatments not just improve behavior in due course, but actually are manifested by changes in brain circuitry that are going in the right direction or not, that would be great news. Can neuroimaging deliver on that kind of, I won't even call it a promise, it's a hope, fingers crossed, but I would not bet the farm that it's going to work, be spectacularly successful. I think there will be important progress, but nobody, including the public, should underestimate the staggering complexity of the human brain, healthy and diseased, and the challenges we face to really grapple with these problems. Just exciting, but very challenging. And the patient community is clamoring for action, and everyone's working their hardest, but we have to realize these are deeply hard problems.
Tony gets the last question.
So, just to take a step back from actually intervening, say with your note of optimism about applying this to disease, what do you think the scientific obstacles are to using, say, a resting state scan for diagnosis of mental disorders?
I think the practical, when you say scientific obstacles, I think the most...
Plenty of political ones.
No, I think the technical obstacles are patients are less patient. You can't scan for as long. You can't get 4 hours worth of data like we do on healthy adults. And they're more wiggly. And those movements in the scanner create what are now more broadly appreciated as artifacts that need to be adjusted for, and be careful not to overinterpret your data. So, you know, the resting state, the functional brain imaging is telling us vastly more than we could have imagined a few years ago. And in some ways, it's telling us less than we had hoped when the vision of, you know, millimeter level resolution and exquisite signal to noise. You're engineers. The realities are, what's the quality of your signal, and how do you zoom in on the signal and not get distracted and obscured by the noise? And these are hugely important methodological, technical challenges for the field to collectively wrestle with.
Okay, let's give our speaker another hand. So one of the really nice traditions that Alan and Constance Ford have brought to this lecture series is that each one of our speakers gets an original piece of art. So I'd like to present this to you, David. This is from Erica Weiss from the Juma Gallery, which is just up the street here in Little Italy, as a token of our appreciation for joining us today. So thank you.